opportunity to speak today. So whenever I heard that this was about machine learning for pathologists and sort of a realistic view of machine learning, I made the fateful decision that I found that really interesting, so I thought I'll make a new presentation, which this morning didn't feel like such a good idea. So some of the slides are going to surprise me as they come up as well. But I'm going to speak mostly about QuPath, and which is the software that I developed, and illustrate the concepts of machine learning using that. And a bit more on the classical machine learning as opposed to the deep learning, but there's going to be a bit of both. Okay, if I can. Okay, so I want to begin by speaking a little bit about the general automatic image analysis pipeline, where of course you've got a black box in the middle. You've got the images going in, and then some kind of stuff happens, and eventually results come out. And so the focus of this talk is going to be mostly what's going on there in the middle, and I'm going to try and give a lightning tour of image processing, which is going to reiterate some of what Jeff had said. So in this part of the workshop, I want to convince you of a few things. One of them is that image analysis is not free from bias. So it's often said that we applied automated image analysis, so it was unbiased. I don't think it is. I think there's all sorts of scope for biases, but some biases are worse than others. But image analysis should be reproducible, and if it's not, then generally something's gone badly wrong. So if you've ever forgotten to seed your random number generator, you might recognize this illustration. So it should be reproducible, but really we want algorithms which are reproducibly useful, and that's where we need to understand one another. The people who are developing the algorithms and the pathologists being able to talk and communicate. And I would argue that software is one of the things that really helps us do it, and the visualizations help us do it. So if a pathologist comes to me and they say they want to find an area of dysplasia, I'm not going to find the right thing, but if they can actually show me and illustrate and annotate, and I can show them what the algorithm's doing, we might be able to come up with something useful. So my background's in bioimage analysis kind of broadly, with a lot of fluorescence microscopy and a little bit of retinal image analysis. But I kind of developed my views on image analysis mostly while I was a postdoc at Heidelberg University working in a core microscopy facility. So there we had a lot of users of the facility with lots of diverse problems needing an image analysis solution, and they were not maybe so into the percentage accuracy, they were into the fact that it gave them the right results. And so while I was there, I wrote this image analysis handbook because I found that I was using a lot of the same tools and a lot of the same techniques repeatedly, and so I thought I could write them up, and I thought it would be about 20 pages, but it ended up being considerably longer than that, and so this is all freely available online. And during that time, I depended pretty much exclusively on open source software to solve the problems. So at the end of 2012, I came back to Belfast and joined Peter Hamilton's group working in digital pathology, and I was there for 4 years as a postdoc, and I spent the first 2 years trying to apply the existing open source tools for digital pathology and to work with these whole slide images. And I developed a number of different bits of software, and one thing they had in common is that they were horrible to use. I didn't like using them, nobody else ever did use them, but they were not the most user-friendly or pleasant things to work with. And if you needed to customize them, you were kind of stuck because they lacked the kind of interactivity and visualization tools that you need. And so then I spent the next 2 years as a postdoc working on QuPath. So at that point, after 2 years of doing stuff that I wasn't very satisfied with, I started to think about what is the platform that I wanted to use for digital pathology, and ideally wanted to be open source, and so that's what I set about then developing. So this video is showing in double speed. It normally takes 5 minutes, so it's going to take 2.5 minutes in the interest of time here, but this is showing QuPath as it was whenever I left that postdoc position in Belfast. And what you're seeing is one of the applications from Belfast where we've got a whole slide image of a tissue microarray. It's about a 40 gigabyte image. We've got 90 breast cancer patient samples within it. And with QuPath, we've got the whole slide viewer. You can watch the processing happen now. There's an automated de-arraying algorithm that I developed. So we've identified the 90 breast cancer samples, imposed the tissue microarray grid. Now I'm running cell detection within QuPath on that, which is again another algorithm developed specifically for the software. And it's detecting about 300,000 cells across here. And so the tissue microarray samples are stained for estrogen receptor. You can see that as the cells appear, some of them are red, some of them are blue. It's actually detecting the nuclei, expanding them to approximate the full cell, and making 33 different measurements per cell, and then classifying them based upon the nuclear staining as being positive or negative. So if you only want the percentage of positive cells, then you would be done. But ideally, we would then be getting the percentage of positive tumor cells. And there we can start to take advantage of the fact that we have all of these additional measurements. So I think visualization is really important to know what's happening. So I created visualization tools that allows us to see the measurements, to supplement them with additional contextual measurements, for example. So here we've got the nucleus to cell area ratio, and then I start to train a machine learning classifier, which I am totally unqualified to do because I wouldn't be necessarily the best person to distinguish what's a tumor cell or not. But that's what I'm doing here. I'm giving it some examples, and it's using interactive machine learning to train a cell classifier based upon what I'm telling it. And so red indicates it thinks it's tumor based upon my annotations and all these feature measurements, and then green, it thinks it's non-tumor. Whenever I'm relatively happy with the performance, I then subclassify each cell according to the staining intensity. We can get a summary H-score of all the tissue microarray cores across the slide. We can rank them if we want, and if you happen to have survival information, you can actually copy and paste that into QuPath. And so from opening the slide, to detecting the cells, to generating a custom classifier, to generating survival curves at the end takes 5 minutes in this case, which I think is pretty fast for this application. And QuPath will also store all of the steps that you did. You can generate a script, and then you can apply it in bulk across multiple images if you need to. And so this is what the software could do at the end of my postdoc. It was not the easiest thing to convince journal editors that this was worth publishing, but eventually it got published in scientific reports. And it's become one of the most widely used digital pathology platforms. I'm not really sure what the statistics are for other software, but it's got more than 30,000 downloads. I've taught some workshops in Europe and North America. It's used in 80 publications, including in Nature and Cell. But it's not just the software. So in addition to writing all the code, I created blogs, user forums, and all that kind of stuff. And so there's YouTube tutorials, there's all this built around it, and there's more and more people engaging with the software now. But the most interesting publication for me is the one that I had nothing to do with and didn't even know that it was happening, which was this sort of independent comparison of different digital pathology software from David Rim's lab at Yale, where they concluded, after comparing QuPath with other commercial platforms, that there was outstanding reproducibility within and between the digital image analysis platforms, including with QuPath, and they were essentially indistinguishable with respect to prediction of breast cancer patient outcome, looking at Ki-67 in breast cancer. And so the fact that it's free and open source doesn't mean that it's necessarily inferior. So I left Queen's at the end of 2016, and then I wasn't really working on the project over the next year or so, but in September last year, I joined the University of Edinburgh to restart work on QuPath, now as a senior lecturer. And last month, the first new milestone version of QuPath was released. It's called Milestone because it's not really finished, but I was going to the US and I wanted to talk about the new stuff. And so it's still a work in progress, but based upon the feedback that I'm getting, I'm fixing bugs and improving it as it goes along, and there's already quite a lot of new features within it. And that's what I'm going to talk about mostly today. So one thing I do feel I need to make clear is that QuPath it's not an algorithm. So it's not the case that you have one particular application that you would run QuPath. It's not a tissue microarray analysis software or a tumor identification software, but rather it's designed to be a software platform that you can customize it and do interesting things with it and solve all sorts of different applications. And so the interesting stuff isn't what QuPath itself does, it's what people use it to do. And so the aim is to make quantitative analysis more rapid, reproducible across many research applications. I aimed it towards pathologists and biologists. I've got more of a computer science background myself, but I created it because I needed a way to create algorithms myself rapidly and to be able to share them with others. And so it's made for image analysts and algorithm developers like myself. So then, relating this then back to machine learning, I want to show the machine learning concepts through QuPath as the example. And I want to begin by taking the simplest application that I could come up with, which was quantifying a stained area. And then we're going to look at positive cell counting. And so this, you can apply QuPath to H&E, fluorescence, and all sorts of other things, but I've chosen these two examples not because they're necessarily the most exciting or clinically relevant, but because you can actually download these images. They're freely available through the fantastic OpenSlide project, and so you could, if you wanted, look at them and apply the same ideas yourself. And so we've got one image where we want to quantify the stain area and one where we want to count the percentage of Ki-67 positive cells. So beginning with this simple application of the stained area, I would like to give you an overview of image processing and show how you would approach this without machine learning, and show that despite the fact it seems very simple and well defined, actually there are complications in there that need considering, and then show how these relate then to machine learning. So to begin, images are composed of pixels, as we saw and probably all know, and whole slide images have a lot of pixels. So here we've got about 10 billion of them. It's about 30 gigabytes of data. If I select a region, I can expand it, and you have to expand pretty far before you can start to see the individual pixels within it. But here, these little squares, each square corresponds to a pixel, which is about 1/4 of a micron in size. And as Jeff said, the image then, these bright field images, they're RGB, which means they've got a red, a green, and a blue channel. And so each pixel has got 3 numeric values, one for red, one for green, one for blue. And because these are just numbers, we can start to do interesting stuff with them. And so, for example, we take the pixel values and we just replace each pixel by the average of the surrounding pixels. And then that results in a smoothed image. So Jeff mentioned convolution. That's essentially what it is. So we've smoothed an image, and if we average by more pixels in a larger neighborhood, we smooth it more. And so we might want to then subtract one for the other, and then we get an image that might in some way be useful. An alternative is we might take the pixel on the right, subtract the one on the left. That gives us a gradient image. We might do the same vertically, combine them, and then we get an edge-enhanced image. So all of these kind of approaches, that's basically image processing. You take the numbers, you do something with them. Very often it's basic arithmetic, but the fact that you have, in this case, 10 billion pixels, they start to make some real differences. And a common operation that you would apply is to computationally separate out the stains using color deconvolution. So you can see we've got a blue stain, brown stain, but you've got red, green, and blue channels. And so these channels don't match perfectly with the stains. But the algorithm called color deconvolution means that if you can characterize the blue and the brown stains, you can computationally separate out the channels in the image and then apply them to get some kind of measurement of the staining intensity. It's not terribly quantitative, but it's still practically very useful. So then, if we want to look at the stained areas, we can apply color deconvolution to our image, and then we can apply a threshold and say, if the pixel value is above a certain threshold, then we consider it to be positive for the staining. But the threshold is really key. So I've given 3 examples here where if I choose a low threshold, it's maybe almost 50% positive within the region that I've shown, 18% positive with a moderate threshold, 8% positive with a high threshold. And so there's a question of when is a pixel really stained enough to count? And so how you determine this threshold can have a massive impact upon your results, and you could choose different thresholds and get every possible value in between. But this isn't the only decision that we need to make with this simple example. We generally don't want to process 10 billion pixels if we don't have to, because that's going to be pretty slow. So we can take advantage of the fact that these whole slide images are stored at multiple resolutions. So we have the same image data at different resolutions. That's the so-called image pyramid. And if you have software that can written to handle this, like QuPath has, the software will go in and pull out of the pyramid just the part of the image that it needs at any particular moment. That can be for visualization, but it can also be for analysis. And so if I am to work at a high resolution, so each pixel corresponds to a very small area, then I can get a very fine grained kind of identification of the stained pixels. So here, a very fine resolution, but it's going to be slow to process in that level of detail. And if I change the resolution, you can see that it will impact the accuracy of the regions I've identified, and that will impact the stained areas that we generate. And so despite the fact that this is a really simple application, and it's hard to think of a much simpler one when it comes to quantitative analysis relevant to pathology, you still have to make some crucial decisions. You have to choose the resolution of the image to analyze, the stain colors, how you separate them, how you characterize the blue and the brown, and the choice of threshold. And you can get a wide range of different results by approaching these in different ways and making different decisions. And so you might wonder, can machine learning spare us all this decision making? And I would say it can't really, but it changes the decisions that we need to make. And so, for example, if I take this image and I approach it from a machine learning point of view as opposed to separating the stains and thresholding, then I can start to annotate regions and I can point the system in the direction of what I want it to detect. And then I apply some kind of classifier. And it could be a deep neural network, as mentioned before, but it could be a decision tree, as was also previously mentioned. In that case, here I'm showing a decision tree where essentially what will be learned by the machine is for the red, green, and blue values. If red is above a certain threshold, then go down one branch of the tree. If it's below, go down the other, and then eventually reach a decision as to whether a pixel is positive or negative. I could use a k-nearest neighbor approach with machine learning, where when I get a new pixel, the algorithm will then check and find out, is this more similar to what I've seen as positive or negative? Or I could use a neural network where it's calculating these kind of higher level features across the image. And all of them are different sort of flavors of machine learning applied for the same application, and you may get similar results or different results. But critically, you have to choose your approach to machine learning and your training data going in. And so what I'm going to do now is I'm going to show one of the new features from QuPath, which allows you to explore that and to apply it in practice. And so I'm boldly and perhaps somewhat unwisely going to try it with the milestone version of QuPath, because it's not actually in the one from before. And I'm going to show end to end. I'm going to create a project, and that's going to store my images. So this is QuPath, it's the user interface. I'll just drag an empty folder on top. It asks, do I want to create a project? I'll say yes. And then I will try and find where my files are. I said they're from the OpenSlide dataset. These are the ones that I need. So I'll drag one on top. You can see it's added to the project. I'll drag the second one here. And so I can double click on them to switch between them. Okay, and so QuPath, it has a whole slide viewer. I can zoom in, and there's some tricks. So for example, if I want to computationally separate the stains, I just type a number. So the 2 is going to give me hematoxylin, 3 is going to give me DAB, and I can improve the stain estimates if I want. Actually, for the purpose of this demo, you can script QuPath, and so I've written a script, and that's not the one that I wanted to run. I've written a script which will show you all the shortcut keys that I'm pressing in the bottom corner. Okay, so in this case, we want to train the system to be able to distinguish between the different stains. So I can use the annotation tools within QuPath, which are up here, and so these are documented online. I'll select a region. Actually, I'm going to be a bit bolder. I'll use the wand tool, and that'll select the region a little bit more nicely. And I need to assign it a class. So in this case, I'm going to right-click, and I'm going to say, I just want a positive and a negative class. So there are some defaults there that you can see already, but if you want to change from them, you can. So you can add your own classes. So I'm going to say this is positive, and I'm going to annotate a different region, and I'm going to say that is negative. So now, I'm going to use a shortcut to bring up the pixel classifier, and I'm just going to turn on live prediction and see what happens. And based upon the examples that I've given, QuPath will immediately start to classify the image. And actually, I don't really want it to include the white space, so I'll zoom in, I'll just give it a little region there, and I'm going to say ignore that. And I can find the areas where I think it's got it wrong. I can say ignore a bit more. And then you can see that I already have a pixel classifier trained using machine learning based upon the annotations that I've got. And using the shift and scrolling up and down, I can change the opacity to get a view as to whether or not it's done what I want. I can draw a region. And if I look over here under the Annotations tab, it will be able to immediately tell me the percentage of positive pixels in there, ignoring everything that's white space. And I can move it around, and I can get these results. But remember that I mentioned that you could analyze at different resolutions. And so, for example, we're running at a fairly high resolution here. I can make that lower and train up a new classifier so quickly that I can immediately get the results then at different resolutions. And so we have about 50% positive pixels here. I can go to higher resolution, and you can see that the resolution is making a difference. But because you can train a classifier like this so quickly, I have the opportunity to go in and then decide that actually I might want to add some additional annotations and then see, depending upon what I actually want the classifier to do. So if I'm going to say this is negative, you can see it's given me less, but I think it's gone a bit too far, so I'll turn on the magic wand and I'm going to say I want this to be positive. And so because it gives you this instant feedback, you can train it to do what it's supposed to do. And it will also then show you your balance of the training sets so that you know that if you've overtrained for one particular class, you can start to try and balance it out so that it's seen as many negative examples as positive examples, roughly. And so that can help to improve things as well. But because you're working with machine learning, as opposed to using the old separate the stains and threshold, you can actually start to add in additional information. So I can start to smooth the image more, give the classifier a bit more information. I'm going to just work on the annotations. And I can say I want it to also know what a smoothed image looks like and give it all of these different versions of the image that have been processed in different ways. And then it will use that additional information to make its decisions. And if I want to see what it's actually doing, I can then click in here and it will show me the features. And it will show me that for the center of the image, it's taken the image and it's processed it in these various different ways to give the algorithm, the machine learning algorithm, more information. And then it can potentially make a better decision. It might make a worse decision. So you need to be selective in the kind of information that you give it, but you can guide it in the direction of having the sort of features that you would like it to have to make the right kind of decision. What you can also do is you can change the type of classifier, so we can switch to a neural network if you want, and you can choose even the number of hidden layers you might want to have within your neural network. And so hopefully the defaults would work for a lot of applications that use a random forest classifier, but potentially if you want to get deeper into this, the way QuPath is designed, it's for people like me who maybe want to get a little bit deeper to investigate different classifiers, and so that functionality is there, and because you can script it, you can start to explore in new and different ways. So to summarize then that, you can see that for quantifying stained areas, even this simple analysis requires making some key decisions, and somebody has to decide them. And so if you're using a software where you just press a button and you get your result, then somebody has made that decision. I guess it's probably going to be the person who developed the software. And since I'm the person developing QuPath, I don't want to be the one who decides that for you, because I'm not a pathologist and I don't know what image you're looking at, what biomarker you're looking at, what you want to do with it. And so from my point of view, within QuPath, I try and give the features and try and document them and explain how they work so that the person who really understands the domain can adapt the software to do what they need. And so this really determines the actual question being answered. And so one example that I looked at, which I haven't shown here, is looking at picoseries red and looking in and liver images to quantify there. And my question for the pathologist is, how red does a pixel need to be? What kind of detail do we want? And I can just try and create the tools that allow you to investigate the image in whatever way is most appropriate. But ultimately, for different applications, that might be the decisions are different. But the decisions do impact the results. And I would say there's arguably no right way to do image analysis, but there's very many wrong ways to do it. And I aim for the least wrong way myself as my goal. And I think that understanding of visualization is important. So I would like the people who understand the data to be able to see what the software has done and then make a call as to whether it's done the right thing. And ideally to interact with it and adapt it as well. So moving on then to the other application, which is positive cell counting. This will show machine learning in a slightly different way. So this was previously in QuPath. So the pixel classifier is entirely new within these recent milestone versions. But the cell classifier has been there since the start. And so essentially here, we're going to want the percentage of positive tumor cells. And again, I am not the most reliable person to say what the tumor cell is, but we can use the brush tool within QuPath. A nice feature about that is depending upon how far you zoom in, you can either annotate in a very detailed way, or you can zoom out and annotate a large region. You can hold down the Alt key to subtract, and you can press Shift and F to fill it in, or not. And so the annotations in QuPath are quite fairly comprehensive because I needed to annotate a lot myself. And generally, if I had to suffer something a lot, then I probably wrote a command to try and help. Sometimes users suffer a lot, and unless they can give me a project where I have to suffer too, I might be slower to implement something to fix it for them. But there is then a positive cell detection command within QuPath, which I've just run here, and it will then try and identify the cells. It was run in the demo earlier, identify the nuclei, expand to approximate the cell, and then give you a count of the positive and negative cells within that region. And so I suspect that with these settings, it's going to be fairly unconvincing in some ways. And I've got a YouTube video that describes how you can optimize and how you can understand these various different parameters. So this isn't machine learning. This is an unsupervised algorithm where I give you lots of options, and the options that you choose will influence what the algorithm does. So I'm going to change this one, which is going to detect on a slightly different image, and I think it's going to clean it up a little bit. But what you can do, one of the key things, as I say, is the visualization. So you can see what's detected, you can double click on a cell, you can view its measurements, and then you can start to adjust the parameters and optimize them if you need to. But something I never know is when is brown enough to be a positive cell? And so within QuPath, you can adapt this threshold. And so if I increase it, you can see it needs a more intense brown staining for a cell to be called positive, as if I don't. And because you can immediately visualize the results, you can see when you're adapting parameters and not actually influencing the result very much, and when it's really on the threshold of what's important, and then you really might want to spend more time optimizing. But I still think it's probably, in many cases, a good idea to run something like the cell detection in predefined annotations where you define exactly what you want to measure and then you measure it. But what you can also do is you can give QuPath a much larger region and then try and get it to distinguish the different cell types within that. So I'm going to run a cell detection across this larger region. Because the region is quite large, it's going to have to, QuPath will break it up into 9 smaller squares, and then it will apply the cell detection in parallel across this region. And so that's what you can see is happening at the moment. And it tells you how many thousands of cells it's detecting in each of these tiles. And as it does the detection, by default, it automatically makes the measurements as well. Okay. So if I zoom in, you can see that we now have the positive and negative cells, but it has made absolutely no effort whatsoever to distinguish between tumor and non-tumor. But if I double click on a cell, you can see the measurements that have been made. So in the same way as I did with the pixel classifier, I can start to give some examples. And I'm going to hopefully give it somewhat plausible training examples. Go to classifier, create detection classifier, auto update. And then again we have red for tumor and green for non-tumor. And you can adjust these colors if you need to. But you can further then decide that I want to look at the DAB staining in the nucleus, apply a threshold, and then it becomes blue as tumor negative, red is tumor positive, and then this very light pale green is non-tumor. And you can see that I don't know how convincing this is. It looks to me that it's probably made some mistakes, but I can start to then give it additional information and train it further. And just like with the pixel classifier approach, it will then, with auto update, it will interactively train a new classifier. And if you want, you can train it across multiple images, even if necessary. But what you can also do is you can start to interrogate what exactly is the information it has been working with. And so what you can find is we have these 33 different measurements, or actually more in this version. I can view them in a color-coded sort of way. And you can see nucleus to cell area ratio tends to be fairly informative as to whether a cell is tumor or not. So red indicates high values, and then other colors indicate lower values. But at this point, the machine learning has worked on each cell individually. And so based upon the measurement for specifically that cell and nothing else. But what we can do within QuPath is we can say, I want to add some additional information. So we could perhaps look at local textures, Harlick textures, and so on. But a fast trick that I like to do is to just add smoothed features. And so for every cell, QuPath will calculate a weighted average of the corresponding measurements of the neighboring cells. And then we can visualize that. And you can see this is now the nucleus to cell area ratio. You can see that it has sort of cleaned things up a little bit. If I now click, the advanced options, I can choose to select a larger number of features with all these extra ones. And what you're probably going to find is that the classifier has changed, possibly cleaned up a little bit. Actually, I'm going to increase this a lot further. And you can see that whenever I add in more smoothing. So I'm calculating weighted sums of from much wider regions. I can add that information into the classifier, and you can see that QuPath now starts to train a classifier where neighboring cells are almost always going to be the same class. And so this has probably gone to an excess, but you can, by adapting the features going into your classifier, in addition to adapting the training samples that you give it, you can get different results. And so, although I'm just demonstrating these concepts with a random forest classifier, they still apply in general. The kind of network of whatever kind of classifier you're working with, the kind of information it has available, and the training samples all impact the result. And so this is why I want to make the case that image analysis isn't unbiased. There's plenty of room for it to be biased, but you can try and bias it in an informed way and try and make less worrying biases. By understanding what's happening and by understanding the domain, you can make smarter decisions as to what actually is included within the classifier and train better classifiers. Okay. And so machine learning can be applied to objects, not only pixels, and so that's what I showed there. And I should actually close down QuPath so that it gets rid of that little shortcut thing. Okay. So then, finishing up, I want to say a few words then on the elephant in the room, as I haven't really mentioned deep learning or discussed it, because all this is sort of separate from deep learning. So deep learning shifts where the decision making is made, but I would say that visualization and validation is as important as ever. And QuPath itself can also be used to help develop and deploy deep learning algorithms, even though it's not something that is conventionally seen as being able to do. And so one of the easiest ways, or one of the ways in which QuPath is probably most commonly used for deep learning, is as the annotation tool. You saw the magic wand. There are some now additional tricks in there that you can press shortcut keys and it will snap annotations so that they don't overlap. And so you can generate really accurate annotations very quickly using all of these tools and tricks. I also wrote a script which will connect it with a Jupyter notebook so I can exchange information. So I've essentially got a whole slide viewer as a Jupyter notebook along with Jupyter notebooks. And I've written some scripts in my blog showing how you can export annotations in a deep learning friendly way. This is an incredibly old image, but it's showing a deep learning network for nucleus detection trained very quickly on a very small number of examples, but trained using Keras and then viewed through the pixel classifier within QuPath. And so it's a very old example, but just to show that I'm trying to make the pixel classifier in such a way that it is deep learning compatible and it gives you access to all the same tools. But there's more to QuPath than machine learning. You can work with highly multiplexed images, for example. So here's an example of a multiplexed ion beam image, where within QuPath you're able to visualize all of the channels simultaneously. So there's about 44 different channels here. It's very hard to interpret that if you view them all merged. But with QuPath you can move your cursor over and you can now see every channel side by side if you want to. And you can turn channels on and off, and you can adjust the resolution. And this is made possible by the bioformats library, which is able to handle this file format. So bioformats, I think, was mentioned earlier, but it's fantastically useful and important for providing access to this kind of image and data, because QuPath doesn't get into dealing with the difficulties of different file formats itself. Another tool from the same group is Omero, where my video is not playing. But essentially this becomes an image, online kind of image hosting, where you can open an image in a web viewer, you can view it within Omero, and you can actually copy and paste the URL now into QuPath, and you can begin to work with your image. Even though it's stored remotely, QuPath can handle it through this Omero web API. Because it's scriptable, you can begin to interrogate your data whatever way you want. You can start to look at spatial measurements. I've written some posts about that, but it's all... Basically, everything should be possible, and throughout research projects, more and more will become user-friendly and easier to use over time. So if you want to try it, this is the URL. You can download it. If you search for QuPath, there shouldn't be too many other things coming up. And then if you want to get help, there is now this forum.image.sc where there's lots of open source tools represented for image analysis, and now QuPath is one of them. And you can go there and ask questions. And often, I'm not the best person to give you an answer, because if I'm not happy with something in QuPath, I'll probably change the code, whereas you're probably better asking users who don't do that how they've solved the problem. and I'll change the code very slowly, so it will annoy you if you have to wait for me. But apart from that, I want to thank, with open source tools, it's kind of an ecosystem. So there's the bioformats library that I mentioned, there's OpenSlides, there's all of these different kind of components. And so QuPath uses pieces of them, but it is itself a separate piece of software. And so it's written from scratch, but it does then benefit from the ability to integrate with bioformats, OpenSlide, OpenCV, and so on. And to Yvonne, my partner, who's tolerated me talking about this way too much, I would like to thank my group at that point, except I don't have a group, so I only started in September, and so it is still just me. And so it would be good to change that. So I'm advertising a position at the minute, so if you're a software engineer or you know a software engineer that you want to come and join and be thanked on that slide as opposed to just me, then look into this. So the deadline is 22nd of April, and so this is going to be the first position which is dedicated to working on QuPath, because for me it was kind of a side project that got very, very out of hand, and I'm returning to it now. And that's really what I wanted to say. And if there's any questions.
Thank you so much for the fantastic presentation. Do we have any questions from audience? Oh, yes, we have.
Thank you for the nice talk. So I have two questions. The first one, so you almost answer it. You are alone right now working on the product, on the QuPath. So you don't have yet any backup in case of something fails and you are not in to fix it immediately.
That's I don't know how scary to think of that, but yeah, well, it's open source and it's online, and it's up to somebody else to figure out how it's doing if something happens and takes me away from it. But yeah, it's just me. Hopefully that's not going to be the case for too long. So I joined Edinburgh with a PI position with a chance to try and build a group. And so that's definitely the goal, but it does take a bit of time and, I guess, grant funding to actually do that.
Okay, and second question, do you use a high-performance cluster to delegate all these things, or
no.
does this run on the local machine?
Everything I showed you there was running on the laptop. So the initial video was running on my old MacBook Pro, which is quite a few years old, and everything else. So it's designed as a desktop application. You could potentially run it on a high performance computer. Well, you can sort of run it from the command line, but I haven't really focused on that so much because I want to have it, you can download it, you can run it locally, and you can do most of what you want to do. Because I think that's kind of what's missing, and there's more of a need for that in this field at the moment, or at least there's not other solutions for that are open source. So that's what I focused on. You can run it locally, and it tries to be efficient. Ideally, you've got enough RAM.
Is it Java-based or
yeah, Java FX for the user interface. It's Java-based, but it uses OpenCV for some things, so you can sort of access C++ libraries and so on if you need to, but it's Java itself.
Okay, thank you.
Thanks.
Yeah, we have time for one more question, please.
Thank you for a nice talk, and I was using this software yesterday, and it had plenty of functionality. So I'm just wondering, like, is there any way to export the segmentation results? Like, usually we can, there is a way to export the quantitative result, like cell count, but I'm wondering about the cell segmentation, like for the whole slide image.
So because it's scriptable, you can really export whatever you want in whatever way you want. And so, yeah, so I've written some export scripts that will export the coordinates. You can export segmentation masks, whatever really you need to, but it probably needs a custom script. If there's a really common method of export that should be standardized, then it could be added into it. But at the minute, I don't know what would be the commonly used method of export that is necessary and worthwhile integrating into it. And so this kind of thing would still be taken care of with a script. But there's helper functions within here. So within QuPath, you can write a script that it has its own way of representing regions of interest to keep it small and concise and not use up too much memory, but there's helper functions to convert it into Java topology suite geometries, Java AWT shapes, ImageJ ROIs, and so you can convert into these different formats if you need to, because it tries to be simple enough to make it possible to switch into whatever representation you require.
All right. Thank you.
Thanks.
One last question from me, Pete. Have you ever thought when probably you thought, obviously, in which direction QuPath should go in the future, because you are wanting the team to grow actually. But have you ever thought for making it available to the daily pathologist's routine? It would be somewhat important to incorporate the software into something else, maybe into a LIS or into viewers of other companies from other vendors for host-light images, for example. Have you ever thought of that in that direction?
Not a lot, but then that's the sort of thing that's, yeah, if somebody wants to have the conversation, I can talk about it. Me thinking about that on my own, I'm not going to get very far because I don't know enough about what's involved in that. My interest is mostly in terms of maximizing the usefulness of the software. So it's not to maximize the users and to see it everywhere. It's to try and fulfill the areas where there maybe isn't a solution to do it. And so I don't know if it's the best fit in there, but I'm interested in having the conversation. But yeah, my goal is to maximize the usefulness rather than see it everywhere. But if it makes sense, then it's worth talking about.